Good morning, everyone, and welcome to the Center for Iron and Heme Disorders virtual seminar series. Today, we are so happy to have Dr. Hongying Shen come and speak to us from Yale. I want to introduce her in just a minute, but I want to take a second to share something with you that maybe is too near and dear to my heart, but I'm going to show it anyway. First of all, this is our website for the Center for Iron and Heme Disorders here at Utah, where you can go learn about core facilities and activities that are going on and funding opportunities and seminar series. And I hope you'll take a chance to go look at that. One of the things I wanted to guide you through here is two links. One is this opportunity right here for trainees in your labs to potentially come to Utah to train at one of the Center for Iron and Heme Disorders cores. We had a call that was due November 1st, but we've extended that to the end of November. So if you have a trainee in your lab who might be interested in coming, it's actually an event that occurs in March. We have a symposium with a keynote speaker, and then 2 days of training in a core of interest, metabolomics, lipidomics, protein metabolite discovery, iron and heme core, or the mutation generation detection core. And if you're interested in that, you're welcome to reach out to me if you have any questions, or just simply send an application for your potential individual or trainee. And then one other thing, the CIHD has a YouTube channel in case you haven't checked that out. And if you go in here and click on that YouTube channel, you'll find several different playlists from different years of these virtual seminar series. This year we did something unique, and we have done interviews with giants in iron, is what we call it. And the playlist includes 5 different individuals so far, including Harry Daly, Nancy Andrews, Roland Lill, Jerry Kaplan, and Tom Gantz. And these are kind of a fun history of how these individuals got into the field of iron and sort of their memories traveling down the iron venue. and they're fun. They're super fun to listen to. And I know Roland is on this, so thanks, Roland, for participating. And if you want to kind of go and hear some history about iron metabolism, these 5 individuals can tell you some unique stories, and that's very fun. With that, I'm going to stop sharing, and I'm going to just introduce Hoi to you. Hoi comes to us from China, where she got her bachelor's degree from Nanjing University and the bachelor's degree in chemistry. She then went on to a Ph.D. at Yale in the lab of Pietro de Camilli, studying lipids and membrane trafficking, something near and dear to a few people in this meeting's heart. She then went to Vansi Mutha's lab, where she worked on mitochondrial physiology and high-resolution mass spectrometry metabolomics. And today she's going to talk to us about some of her discoveries on a particular metabolite transporter and some surprising discoveries with the connection with iron metabolism and glutathione. And thank you so much, Dr. Shen, for speaking to us today. I'm going to ask everyone mute themselves. If you have a question, you can populate it in the chat, or you're welcome to unmute yourself and then ask the question, or I can invite you. Any of that works for us. And with that, we'll turn the time over to you. Bye. Thank you.
Thank you, Diane. Thank you, the Center, for the invitation. I think I view this opportunity more as a chance for me to learn more about iron metabolism. So please feel free to stop me, ask questions, or correct me if I make mistake.
Okay. Looks good.
Awesome. Yeah, so I slightly changed my title a bit, given the audience and also the special interest on glutathione and iron metabolism inside the mitochondria. So the lab study mitochondria membrane transporter, and what we actually get into this problem is actually unexpected discovery related to iron, and then you will hear more about it. So the type of questions that we are particularly intrigued is the idea that human genome actually encoded hundreds of metabolic enzyme and transporters that we still do not know their function. And at the same time, the genetic study in human have already associated some of these enzyme and transporters to human diseases. So despite the lack of understanding on their biochemical activities, some of these transporters and enzyme might contribute to human diseases or have been identified as the hits in forward genetic screens that lead to either cellular or organismal phenotypes. And with my lab's expertise in mass spec metabolomics, we begin to wonder if we can combine that technique with traditional cell biology and biochemistry and begin to first characterize the biochemical activity and provide the initial hints on their function, and perhaps that will guide us future physiological study how these genes might contribute to human diseases. So the processes that my lab currently study is the mitochondria metabolite transporter. So we all know the mitochondria are considered powerhouse of the cell, and it's doing so by being a gigantic reaction chamber that houses a variety of different enzymatic reaction, including electron transport chain, TCA cycle, mitochondrial central dogma, and fatty acid beta oxidation. So to support all these different metabolic reaction, mitochondria need to constantly translocate a variety of different metabolites in and outside of mitochondria, including amino acids, nucleotides, cofactor, iron, carboxylic, and fatty acid. However, different from mitochondria outer membrane that is considered permeable to polar metabolite smaller than 5 kilodalton, mitochondrial inner membrane is considered impermeable to even the smallest polar metabolite proton, and that's actually the reason how proton motive force can be built up and generate the energy charge molecule ATP. So this actually might come as a surprise to me and then maybe the audience. It turned out that the molecular identity for these transporter that constantly translocate metabolite in and outside have not been fully characterized. In fact, the transporters for some of the key metabolites listed here, including calcium, pyruvate, serine, branched chain amino acid, NAD, glutathione, and choline, have all been only been characterized during the last 15 years or so, including pyruvate that was co-discovered by Jerry Rutter, also at Utah. So I think this actually kind of illustrates the importance on the unknowns, how metabolite translocation across the small organelle have not been fully understood. So the largest protein family responsible for that process is called cellular carrier SLC25 family. So there are 53 members encoded in the human genome, and then with exception of 4, majority of them actually sits on the mitochondria inner membrane, and some of the well-characterized ones are known to translocate a variety of different molecules, including proton, phosphate and malate, ADP, ADP exchanger. So those are all enter through the central cavity of this single polypeptide chain generated pool-forming subunits. So we all now know these processes are considered essential. The movement is considered passive or secondary active transporter, meaning they have to move the molecule down the concentration gradient or move one molecule against the concentration gradient by utilizing the other molecule down their concentration gradient. We now know they are monomer, meaning that a single polypeptide chain showing on the right side here is sufficient to generate the pool-forming subunit, allow metabolite translocation through the central cavity. As I mentioned, there are 53 members encoded in the human genome. We still estimate roughly 20 are considered fully characterized function. And then at the same time, the early work from Palmerius and others already associated roughly 20s directly linked to human diseases. So one feature for this family that actually struck us is they are evolutionarily conserved. We can detect SLC25 transporter in almost all eukaryote species. And even in the distantly diverged species, there are at least 10 members encoded in the human genome allow translocating of different metabolites. So this process is actually considered early functional diversification, which means that the same transporter can be derived into molecules that translocate metabolite of complete different structures very early on. And this diversification occurred even before the divergence from the yeast and human, so that when we manually extract SLC25 family members across different model organisms shown here, and then generate the phylogenetic tree, the transporter translocating the same metabolites are clustered together on the same branch of the tree as shown here. For instance, the ADP-ATP exchanger from yeast all the way to human clustered together on this tree that are different from other transporter transporting different molecule. So relevant to today's talk, our transporter is also considered evolutionarily conserved. So there's a yeast ortholog called MTM1, and then the transporter for this family from all these model organisms are also clustered on the same branch of the tree as shown here. And at the same time, in human, it turned out that they are paralogous genes, that the same transporters are actually encoded by multiple genes in the human genome. For instance, human contain 4 ADP-ATP exchangers, and then the A39 transporter we study, there are 2 human paralogs. So because of this functional basic redundancy, what we decided is to perform a screen strategy that we can assess the function of more than one transporter at this time. And in this case, towards the end of my postdoc training, I generate this double CRISPR screen technology so that we can assess the function of the at least 2 transporter in the human cells. And using this approach, we were able to perform the genetic interaction assay between SLC25 transporter to assess both their functional redundancy and then crosstalk between transporters. So for instance, the 2 paralogous ADP-ATP exchanger expressed in the cells exhibit the functional redundancy, that the knockout of the single has little phenotypes, and then double knockout exhibit a severe phenotypes that dramatically reduce the cell growth. In another case that relevant for today's talk, we also observed this buffering interaction in which the A39, a transporter of unknown function back then, exhibit buffering interaction with the major mitochondrial iron transporter A37, so that the single knockout has a little bit growth defects, and then double knockout has no additional phenotype suggesting these 2 transporter actually function in the same pathway. So despite the unknown status for A39, it turned out there are mounting genetic evidence suggesting a potential role of this transporter in iron homeostasis. So first, A39 mutant yeast would find to accumulate iron both in cells and also inside mitochondria, suggesting there might be a role in regulating iron homeostasis. So A39 mutant zebrafish turned out to have defects in heme biosynthesis and lead profound anemia. And then recent work from Kavon's lab, I also identified that A39 mutant mice also have this basically anemia phenotypes that lead to embryonic lethality. So A39 mutant flies turn out to be a forward genetic screen with defects in synaptic transmission and ultimately lead to degeneration of the eyes. So somehow has a connection to neurodegeneration. In human, interestingly, A39 loci have been implicated in epilepsy, autism, and late onset Parkinson's diseases with completely unknown reason. So for that reason, we decided to launch this project to ask two simple questions. What is the function of A39? And then how does A39 transporter protein being regulated? So first, using mitochondrial metabolomics, we were able to assess the metabolic change inside the knockout mitochondria compared to the control. And using this approach, we were able to identify two and only two metabolites that significantly depleted in the knockout mitochondria. These are reduced and oxidized version of glutathione. The impact on glutathione is specific to mitochondria, as we only see glutathione reduction inside the mitochondria for both reduced and oxidized version. We did not observe any change in the ratio, suggesting the redox might not change. We did not observe any effects on the whole cell glutathione level, as shown here, suggesting it's specifically affecting mitochondrial entry of these molecules. Then we perform the mitochondrial uptake assay using stable isotope labeled glutathione. When we purify the mitochondria from control or knockout cells and then incubate with labeled glutathione, we can observe a dramatic reduction of glutathione taken up inside the knockout mitochondria, but not in the control, suggesting the mitochondrial uptake is indeed reduced upon knocking out A39, the transporter. So we can measure that for both singly labeled glutathione, the substrate we provided to the mitochondria, and then we can also detect the reduction of singly labeled oxidized version of glutathione, suggesting that the consequence of this uptake is to combine with endogenous glutathione to generate this singly labeled glutathione. So I forgot to mention glutathione is a tripeptide, and then the biosynthesis pathway only occur inside the cytosol that require this transporter for its mitochondria localization. So what is the mechanism? So the elegant structure characterization for element Hongying slabs have already identified that for SLC25 transporter, it turned out to have a single substrate binding pocket in the central cavity that exhibit the similar distance between the outside membrane and inside membrane. And then upon the substrate binding at this central cavity is triggered the conformational change that allow this binding pocket to be alternatively accessed from either outside or inside. As you can see towards the end of this movie, so the substrate binding were able to trigger the conformational change in and outside. So based on that information, we predict the putative substrate binding pocket, especially the residue, the side chain responsible for recognizing the carboxyl group and amino group that preserved in glutathione due to this double gamma bond. And then we generate mutants that still can be expressed and delivered to mitochondria, but probably will lose the substrate binding activity. And then it turned out that when the white type construct is able to rescue mitochondrial glutathione level, the D2A mutant that we predict cannot bind glutathione directly cannot rescue mitochondrial glutathione, suggesting the transporter is directly recognize the ligand that allowed the translocation. So I have to mention that all the structure showing here is modeled or AlphaFold predicted. Currently, there has not been a structure for SLC25A39, this family member.
Can I interrupt you briefly? Are you talking about oxidized or reduced glutathione?
Oh, that's actually a great question. So in our assay, we test reduced glutathione. We have not directly tested oxidized version of glutathione. However, there's a little bit kind of non-specific oxidation of our reduced glutathione that we did not observe the mitochondrial enrichment. So we actually think the reduced one is being imported, but not the oxidized one. But we are in the process to test that using in vitro system.
Because you may know that there are congenital disorders affecting GSH, GSS, GRH ratio, like glucose 6-phosphate dehydrogenase deficiency. There are also two congenital disorders, gamma glutamyl cysteine synthase and GSH synthase, which cause hemolytic anemia and some other issues. And so the question is, does it affect your system, right?
Yeah, good question. So we have not. So what happened is that mitochondria is actually very reduced. When we look at the ratio between reduced and oxidized version, it's very different from whole cell versus mitochondria. In mitochondria, it turned out that the balance is more shift towards reduced version of glutathione. But that's a good point that we should test that. So what is the consequence of the A39, the transporter knockout? It turned out that knockout of the transporter have defects in mitochondrial bioenergetics, and using seahorse assay, we can detect the A39 knockout cells has reduced oxygen consumption rate under basal condition and also under FCCP treated condition. And they also exhibit compensatory upregulation of glycolysis with the increased ECAR. So that's consistent with defects in mitochondria bioenergetic. And then using the antibody to directly assay electron transport chain subunit stability, we were able to detect the reduction of this a reduced level of both complex 1 and complex 4. And then the reduction in the A39 knockout cells can be rescued to some extent by re-expressing the A39 wild type, but not the mutants that we have demonstrated they are the catalytic defective. And then interestingly, this particular phenotype is also a convergent between A37, the iron transporter, and A39 knockout. So in the A37, the major iron transporter knockout cells also exhibit reduced level of complex 1 and complex 4. And then when we move to double knockout, they seem to be buffering, meaning they are not additive, suggesting both might function in the same pathway. So we don't have the direct evidence, but we believe it's related to iron sulfur cluster. So it turned out that more recently, when we collaborate with Yanxin Liu's lab at Yale to perform proteomics from the knockout mitochondria compared to the control, out of the 45 complex 1 subunits, we can detect it from the assay. We detect 17 are decreased in the knockout cells. And these actually concentrated on the so-called M module, the part of the complex 1 that facing the matrix where the iron sulfur cluster machinery are bound. So we don't have the direct evidence, but with the information we have, we believe that A39 mediated glutathione entry actually worked jointly with A37 mediated iron import, together supporting a process called iron sulfur cluster. And then these are essential cofactor for electron transport chain or oxidative phosphorylation, both for their catalytic activity and also for the protein stability. So then that sounds like an important process. We wonder how A39 mediated glutathione entry can be regulated. So during the study, when we performed the AlphaFold prediction of A39 transporter, we noticed that different from other SLC25 family, A39 turned out to have this extended loop structure. Well, actually, back then we know it's a loop, but we cannot definitively predict the structure. So this is unique to A39 that is not present in other transporter. So this roughly 60 amino acid residues contain 4 cysteines that far exceeds cysteine residue frequency encoding protein. And then initially we thought this is a glutathione, this is iron, this is cysteine, this must be interesting. So we mutate these 4 cysteine and then put that in the cells. The cell is completely fine. They can rescue the mitochondrial glutathione, so we were super disappointed. However, this mutant affects the A39 protein level regulation. So it turned out that the other group study A39 discovered that A39 protein level can be upregulated upon treating the cells with BSO, one of the glutathione biosynthesis inhibitor. And this protein level upregulation doesn't happen at the mRNA, so it can be recapitulated using the flag type Y type A39 and also another mutant A39 with the substrate binding pocket cysteine mutated. However, when we mutate the 4 cysteine in the matrix loop, they completely abolish this protein level upregulation of A39 upon BSO treatment, suggesting somehow this cysteine and the matrix loop is important for protein level regulation. And then another interesting information comes from the correlation between protein level and mRNA level. So this is a omics comparison performed in the HEC293 cell. It turned out that A39 protein level is a protein white outlier, that the protein abundance is much lower than expected when you compare to mRNA abundance. So that data is important, suggesting maybe A39 protein degradation is tightly regulated. So directly assay that, we perform cyclohexamide chase experiment to look for half-life of A39. So under basal condition, pre-synthesized A39 is quickly degraded within 2 hours, suggesting it's a very short-lived. Normally, the mitochondrial protein half-life is considered in the order of 50 to 60 hours. So this is a much shorter compared to majority of the mitochondrial protein. Upon BSO treatment, it significantly increased the half-life of A39 beyond a 5 hours window. So there is stabilization effects. Interestingly, when we look at the 4 cysteine mutants, that was a mutation in the loop structure, both under basal condition and BSO treatment condition, A39 half-life stay low, shorter than 2 hours, showing here. So I think this data is important because it's suggesting it's not a single mechanisms that we are studying. It's actually 2 different mechanisms. In one case, A39 protein is under basal condition, is quickly degraded, and that degradation is independent of the matrix cysteine, the 4 cysteine residue. And on the other case, in the case of the BSO treatment condition, somehow there is inhibition of A39 degradation, so that extend the half-life of A39. And the second mechanism is actually critically dependent on the cysteine residue.
Just a question in there, as you had talked about the regulation and connection between the iron transporter and this glutathione transporter. Did you look at A37 at all in this context? Does it change the half-life of A37?
A37 does not change. A28, we did not find a good antibody to detect endogenous one, but we have the flag tag version, so we can check that.
Okay, there is a good one for 28 that's for human, but not mouse. It works for human. in
Okay.
case you want the endogenous antibody or endogenous protein detected. Thanks.
Awesome. I'll follow up on that. Thank you.
I have a question. So if there is a reduction in iron sulfur cluster synthesis, why are the levels of the proteins in complex 2 and complex 4 decreased?
So we don't know exact. So in the A39, it's a single knockout case. We observe reduction for complex 1 and 4. It is surprising that complex 2 doesn't change, but I think maybe it's due to the exact iron sulfur cluster species. Because when we generate, and also the other group, when we generate the double knockout, basically knocking out both A39 and the paralogous transporter, then all the complex level will reduce. So
Right.
it might be the level
Right.
difference.
Sorry, I misstated my question. I complex 1 and complex 4. I guess my question is that if the cofactor is reduced, why are the levels of the proteins decreased? How does it signal the protein levels to be decreased?
To me, that's also a very interesting question. My sense is that those cofactor are not only important for catalysis, but also a structure component of these electron transport chain subunits, so that they have a tight control mechanisms to sense the level, and if it's not bound tightly, and then it's become unstable and quickly degraded. There has been other mechanisms in protein quality control inside mitochondria that has been shown to affect the level of these complex in a iron sulfur cluster dependent manner. Yeah.
Did you look at other iron sulfur cluster containing proteins to see if their levels are decreased besides electron transport chain complexes?
So we did not do that. Kavan's lab checked the lipoic acid synthesis. There are other processes that also affect it.
Thanks.
Yeah.
Just if I chip in, my name is Roland Lill, and I work on iron sulfur cluster biogenesis. This is a normal situation. You see if the cofactor, the iron sulfur cluster, is missing, then many or even most of the iron sulfur proteins are unstable and become degraded. I usually call it the poor man's assay for assaying iron sulfur cluster insertion.
Thank you, Roland. So to directly identify the mechanisms responsible for these two processes, we perform the immunoprecipitation of the A39 followed by mass spec on the gel. And then we were able to identify several A39 binding partner. So A39 level is very low, and one of the specific measure band turned out to be HSPD1, the protein chaperone inside the mitochondrial matrix, suggesting maybe part of A39 facing the matrix is flexible or unfolded. And then when we analyzed the protein from the whole land, we were able to categorize some of the specific binder into different categories. And then we're using CRISPR to knock out individual hits and assay A39 protein level, both under basal condition and upon the BSO treatment for either 1 day or 2 days. Using that assay, we were able to identify 2 and only 2 protein hits that knockout affects A39 protein level. In one case, it's a protein called AFG3L2. Its knockout dramatically upregulates A39 protein level under basal condition compared to the control, so that it's no longer sensitive towards the BSO treatment. In another case, it's ABCB7. It's related to iron. I'll talk more about it. Knockout slightly increase A39 protein level under basal condition, and it's still sensitive to BSO treatment, can be increased upon BSO treatment. So it turned out these 2 hits are directly related to the 2 mechanisms we are studying. So first, for the AFG3L2, it's turned out to be M-AAA protease that sit on the mitochondrial inner membrane with catalytic site facing matrix. together with a few other mitochondrial proteases, they are considered important for mitochondrial protein quality control to clear out the unfolded protein inside the mitochondria. AFG is also a disease gene, has been implicated in ataxia and Parkinsonian phenotypes, and then the exact mechanisms is not clear, probably due to kind of a large spectrum of substrate has been identified. So in our hand, AFG3L2 knockout dramatically upregulates A39 level under basal condition. So that happened to both endogenous A39 and exogenous A39 for both wild type and then for C mutant. And this is very striking because this suggests that A39 is a specific substrate of AFG3L2. It's probably, I guess, as far as I can tell from literature, is probably one of the best substrates given that level of difference. And when we assay A39 degradation, when the wild type A39 is quickly degraded within 2 hours, the under AFG3L2 knockout condition, A39 degradation is completely abolished so that the protein stay on for 8 hours for the entire assay window. So this suggesting that, again, A39 is a specific substrate for AFG, and the degradation cannot be substituted by other proteases also inside the mitochondria. What is the diagram? It turned out that matrix loops that we initially spotted on is indeed the diagram region. So if we can mutate the A39 long loop to the homologous transporter A40, a shorter loop, or to even another shorter loop from another SLC25, SLC25A11, it's completely abolish this degradation. So as you can see here, A39 protein is quickly is upregulated under AFG3L2 knockout, suggesting it can be digested in the wild type condition. And then if we move to the loop 1 swapped construct, A39 with A11 loop 1, it can be expressed at a high level and no longer sensitive upon the AFG knockout, suggesting this fusion construct is stabilized and is not the substrate for AFG3L2 anymore. And another remarkable feature for this protein level upregulation is that it directly correlates with the function. So as you can see there, with the wild type A39, it can rescue mitochondrial glutathione defects in the knockout cells. This fusion construct that is stabilized under basal condition even increased level of mitochondrial glutathione to a much higher condition, suggesting the regulation directly control the mitochondrial glutathione imports and potentially related processes. So how about the other mechanisms I mentioned? It turned out that the other hits, ABCB7, is another mitochondrial inner membrane protein that has been proposed to export certain iron sulfur cluster species outside of the mitochondria. So knockout of ABCB7 slightly increased A39 level, but not much. And then that seems to be a cysteine-dependent manner. And we thought that seems to suggesting mitochondrial iron is involved in that process. So we decided to directly assay different aspects of mitochondrial iron. And it turned out that that's directly linked to A39 regulation. So first, using a whole cell iron chelator, DFO, we were able to see that the BSO-mediated A39 protein upregulation can be dampened upon DFO treatment, suggesting whole cell iron level is important during this stabilization effects. And then we can directly manipulate mitochondrial iron entry by knocking out two putative iron transporter A28 and A37. And under that condition, it's completely abolish this BSO mediated A39 stabilization effects as shown here, suggesting mitochondrial iron is critical for the second part of A39 regulation. We can also directly assay mitochondrial iron sulfur cluster by generating CRISPR knockout for either HSCB or GLX5. And under that condition, in both cases, actually dampened or reduced this A39 mediated, so BSO mediated A39 stabilization. And I believe there is just some phenotype that's probably due to difficulty to completely eliminate this process because those actually considered essential genes. And to directly assay the connection between A39 and iron, we decided to perform this reactive cysteine labeling assay using this cysteine labeling reagents. And because of the size, upon labeling, they can induce the protein, change the protein migration on the SDS page so that the A39 protein will migrate it to a higher molecular weight. So using that reagent, we were able to detect that under conditions that with the BSO treatment, as shown here, if we perturb the iron level, reduce the iron level, the A39 protein become labeled or the cysteine become freed up under that condition, suggesting there is something directly conjugate the cysteine in an iron-dependent manner. And then the other group studied the same pathway, used the loop structures and able to identify that the cysteine actually can conjugate to 2 iron, 2 sulfur species, suggesting those might be the molecule that kind of, upon binding, inhibit this A39 degradation by AFG. To summarize our work, we first able to identify that A39 is the mitochondrial transporter for glutathione, and this process operates jointly with mitochondrial iron transporter, together support iron sulfur cluster biogenesis and oxidative phosphorylation inside the mitochondria. In the second part, we were able to identify the dual regulation mechanisms. In one case is by mitochondrial protease AFG3L2, and then in another case, the process can be inhibited upon iron sensing. We believe this dual regulation enables an auto-regulatory control that coordinates glutathione and iron metabolism within the mitochondria, suggesting this very intrinsic metabolic crosstalk between these two kind of redox and active molecule. And because of almost every single molecule I mentioned here, shown on the slides, have been implicated in neurological disorder. So the A39 has been implicated in late onset Parkinson's disease. AFG3L2 is a known ataxia and Parkinsonian disease gene, and both iron and glutathione have been implicated in these diseases. We begin to wonder if this regulation might occur in neuronal system, and for that, we decided to use the human iPSC-derived neuron to assay this potential regulation. So in the lab, we were able to derive the iPSC neuron into excitatory neuron. We can detect the spontaneous synaptic transmission. When we assay the A39 protein level, something is interesting. The protein level of A39 actually decrease during neuronal differentiation. So day 21 is considered mature neuron. So it's actually decreased. The mRNA level also decreased, but not as dramatic as the protein level. And it turned out that the those neuronal cells still have this capacity to modulate A39 protein level upon BSO treatment or iron chelation, so that the A39 protein level in mature neurons still can be dramatically upregulated upon BSO treatments, and that can be dampened upon iron chelation, suggesting that in the neuronal cell, despite a low level of A39, it can quickly respond to certain kind of metabolic cue and then allow regulating mitochondrial glutathione in response to iron homeostasis. So we're trying to explore this particular mechanisms in the context of neuronal cell physiology and in the context of brain disorder. So to summarize the talk, we discover this interesting redox compartmentalization and also metabolic coupling entirely start from a transporter of unknown function, as shown here as only 2 small branches on the tree. And then I guess that's hopefully will demonstrate a little bit our approach, how we can start from a metabolic genes of poorly characterized function and then link it to certain biochemistry, mitochondrial glutathione and iron homeostasis. And hopefully one day we can kind of crack open certain part of human diseases and understand the implication in brain disorder. And of course, given the importance of iron in blood cells, and I'm sure there's kind of interesting crosstalk between different organ systems. So for that, I would like to thank my group who did this fantastic work. Xiaojian Shi, a talented postdoc fellow in the lab, just spearhead the 2 work and just it's like just absolutely amazing. It's a rare combination between analytic chemists using the LC mass spec metabolomics and then more detailed cell biology and biochemistry. So the work is also supported by Marissa's in the lab, alumni Katie, Luana, and Richard. So these are my collaborators for various stage of the projects. Yeah, thank you for your time and appreciate your listening.